The Chincha Islands off the coast of Peru are known for their coveted white gold, which helped the Chincha Kingdom rise to power over 1,000 years ago. And when the Incas came to power in the 15th century, it helped them grow staple crops, including potatoes and quinoa. In the mid-1800s, this white gold led to war between South American nations and Spain, and even prompted the United States to seize nearly 100 islands across the Pacific Ocean and Caribbean, several of which are still U.S. territories today. Wars over riches and resources are nothing new, but what was different about this white gold compared to other riches was that it stunk. Literally. Because it was bird poop. Welcome to Tiny Matters, a science podcast about the little things that have a big impact on our society, past and present. I'm Sam Jones, and I'm joined by my co-host, Deboki Chakravarti.
Sam, that was quite a vivid start to our episode.
I'm glad you thought so.
That stinky white gold you mentioned is better known as guano, a word used to describe dried excrement of seabirds, bats, or seals, though we'll be focusing on seabird guano. These birds eat a lot of fish, so their waste is super high in nitrogen, phosphate, and potassium. And because of those nutrients, guano was, and sometimes still is, used as a fertilizer. But despite its historical importance, guano more or less flew under the radar for most of the world until the 1800s, when industrialization was taking hold in Europe and the United States. Populations were dramatically growing and farming was changing, both how people were farming and the crops they were planting.
Farmers had kind of had some concept for a long time that you had to return manure or animal life or other aspects of life into the soil, and there was other techniques such as rotating crops or controlled burns or planting certain types of plants that kind of kept the soils healthy. But this was now going away.
That's Greg Riley, who's part of the operations team at Rich Earth Institute, a nonprofit in Brattleboro, Vermont, whose mission is to advance the use of human waste as a resource.
Guano had built up for centuries in this kind of like concrete calcified form, and it became the solution to basically solve an industrialized farming practice that was not replenishing nutrients to the soil.
And like I mentioned earlier, that push for guano inspired imperialism and war. The United States passed the Guano Islands Act of 1856, which essentially allowed U.S. citizens to claim unoccupied, unclaimed islands containing guano. And that led to the seizure of nearly 100 islands.
We would go for the poop islands.
Yeah, we absolutely would. Meanwhile, Spain fought Peru over control of the Chincha Islands, and Bolivia, Chile, and Peru fought each other over issues that included guano taxes.
And so pressure was on to find a fertilizer solution that was not seabird excrement. And that is the focus of today's episode. We're asking, what is the fertilizer we think of today? How was it first made? What are the implications of its production? And are there alternatives? On a fundamental level, fertilizer is what you need to replace the nutrients in soil that plants take up as they grow.
So if you grow a corn plant, it's going to pull nitrogen, potassium, phosphorus, and other elements out of the soil to build the corn plant as it grows. When you harvest that and sell it as a food product, people eat that material that contains nitrogen, potassium, and phosphorus. It then goes into our bodies, we digest it, it does all the things that it needs to do to nourish our bodies, and then we excrete that nitrogen, potassium, and phosphorus and other nutrients in our bodily waste, and most of that is in the urine.
That's Abe Noe Hayes, research director of the Rich Earth Institute. Their big, overarching focus is urine.
I would love it if everyone realized what was coming out of their bodies every day, that the nutrients that leave our bodies in our urine contain enough fertilizer to grow the wheat you need to make a whole loaf of bread every day. That much fertilizer is going directly into the toilet. That really is enough to sustain ourselves.
Way, way back in episode 64, we gave a nod to more circular systems where human waste is separated and collected before ever reaching a septic tank, allowing urine in particular to be converted into fertilizer. We couldn't get into it more at the time, but the idea of exploring these systems stuck with us.
And then I heard from my cousin, Greg Riley, who came in at the top of the episode, and he pointed me toward the Rich Earth Institute and Abe. He told me about the stuff that they're doing locally and the collaborations that they have with various universities. And it's all really cool. But to really understand that work and why such a focus on urine, we need to talk about an element that many of us take for granted, but that does a whole lot: nitrogen.
And we found the perfect person to talk with us about it. Leila Duman is an organometallic chemist who did her PhD work in nitrogen fixation. She really loves nitrogen.
She really does.
So we had to ask her why.
Okay, this is one of my favorite questions in the world because I think nitrogen is like the most underrated element because it makes up 80% of the air that we breathe almost, but it's also an incredibly useful chemical building block because it lets you make turns in molecules. It lets you have these odd angles and double bonds with single bonds, and it creates chemical geometry that you really can't get any other way. So it's like the foundational building block for things like proteins, fertilizers, which end up building our proteins, a lot of pharmaceuticals, some weapons. So modern life could not exist as it does without an abundance of nitrogen in these chemicals.
Okay, I mean, that is a very good argument for the importance of nitrogen, but nitrogen is also a little tricky. It's a gas, N2 or dinitrogen, and the two nitrogens are stuck really tightly together by a triple bond. Breaking that bond between the two nitrogens and then getting those single nitrogen atoms to bond to something else is the process of fixation.
So my saying is, when it comes to dinitrogen, if it ain't broke, you can't fix it.
Wait, that's so good.
My lab mates were so angry with me the day that I came in the lab and had thought of that in the shower the night before.
Have you trademarked this? We have to trademark it.
I haven't trademarked it. It's in my thesis. I've run it at like conferences before, and it doesn't usually get a good laugh, I'll be honest. So if you take nothing else away, right, just you need to know that it's very, very difficult to cleave those dinitrogen bonds. And in nature, you see it happening like with lightning strikes or volcanic eruptions. Like it takes an enormous amount of energy to do it.
But there's also a powerful little enzyme called nitrogenase that can break those bonds. You find nitrogenase in the bacteria on the roots of legumes in particular, things like soybeans, peas, and lentils. And it allows for nitrogen fixation at not volcanic eruption level temperatures. So remember earlier when Greg mentioned that for a long time, people knew rotating their crops was a good idea? While they didn't know exactly why this was important at the time, rotating crops helped nitrogenase-producing microbes grow really well, which meant they could also convert atmospheric nitrogen into bioavailable fertilizer.
And for most of the world's history, that's all we had was just nitrogenase, chugging along over the scale of the geographic scope of the planet, fixing nitrogen into things that eventually ended up either fertilizing soils or going through bodies and then ending up in things like guano that people used then to fertilize soils that didn't have a lot of nitrogen in them naturally.
It always comes back to poop.
Always.
So for centuries, guano was another source of fertilizer in some regions, but it reached white gold status as populations grew and spread across the globe. In the mid-1800s, there started to be major concerns that parts of the world were going to end up starving because of a lack of fertilizer to grow crops. Humanity needed a new solution. Enter Fritz Haber, or Haber, depending on who you ask, and Haber-Bosch.
It was seen as one of the great challenges of the early 1900s was who's going to save mankind by figuring out how to synthesize nitrogen fertilizers. And the answer ended up being a chemist named Fritz Haber. He was born to a Jewish-German family in the 1860s. And he was a really ambitious and talented scientist. He sort of worked his way up through various German institutions, and he really loved Germany. And one of the things that he took on was this sort of mantle that he wanted to solve one of these enormous crises facing humanity. And through some lucky breaks, but a lot of, ingenuity and work. What he was able to do was to fix nitrogen from the air into liquid ammonia in the lab. And there were some other very, very small scale applications at the time. But his demonstration was really the first time that someone had been able to do it in a way that you could envision scaling up to massive quantities.
Fritz Haber's 1909 demonstration was in the lab, so on a very small scale, as Leila mentioned. In this demonstration, Haber ran nitrogen gas and hydrogen gas over a metal osmium catalyst to speed up the reaction, and it worked. He was able to break the nitrogen molecules, combine them with hydrogen, and create liquid ammonia, NH3. And still today, ammonia is the base material for fertilizers.
So what Fritz Haber did back in 1909 was incredible, but remember, it wasn't on a large scale. That's where Carl Bosch came in. Bosch, also German, was a gifted engineer at the company that had acquired Haber's process.
Bosch was tasked with taking this tiny miracle and building it into something that would revolutionize society. So he did this by essentially having to create a way to turn a couple drops into something orders of magnitude bigger. We're talking tons and tons and tons. Bosch was sort of also known as the godfather of highly pressurized chemistry, because what he had to do, if we go back to our Gen Chem, we know that we can speed up reactions by increasing the temperature and often increasing the pressure. That's one thing that Bosch was able to do. He was able to create these enormous furnaces that were able to have really intense temperature and pressure going on at the same time. He also worked to develop alternative catalysts.
Just a year later, in 1910, Bosch and his colleague Alvin Mittasch found a highly effective iron-based catalyst.
What Bosch was able to do was scale up on an order of magnitudes, lower the temperature and pressure that was required by finding a more efficient catalyst.
And within just a few years, the high demand for guano subsided. Now there was a far cheaper, easier option for fertilizer.
Once they had established the industrial size of this process, they started building factories in Germany. And I think it wasn't until after World War II that the factories really became global, because at that point, the Allies were coming into Germany and seeing these factories and how they operated. But there are some estimates that the fixed nitrogen that the Haber-Bosch process produced was so impactful that it allowed Germany to keep from surrendering in World War I for several years after they would have had to.
Right, because ammonia isn't just super important for fertilizer. Nitrogen compounds are also used in explosives. And more on that in just a moment. Pale Blue Pod is an astronomy podcast for people who are overwhelmed by the universe but want to be its friend. Astrophysicist Dr. Moya McTeer and her best friend Constar demystify space one topic at a time with open eyes, open arms, and open mouths from so much laughing and jaw-dropping. By the end of each episode, the cosmos will feel a little less, too scary, and a little more, oh, so cool. New episodes every Monday. So subscribe to Pale Blue Pod in your podcast app now. That's Pale Blue Pod from the Multitude Podcast Collective. All right, back to the show. So there was another layer to Fritz Haber. Of course, what he did for humanity by figuring out how to fix nitrogen was incredible. But depending on who you talk to, he's often better known as the father of chemical warfare for his development of chlorine gas in World War I.
So if you also want to think about World War I and trench warfare and all of the horrors that came with that, I mentioned that Haber, he had a lot of love for his country, and a lot of what he did was intended to help Germany. And that's one of the things that he did, was essentially create chemical weapons.
If you want to learn more about this, listen to ACS's new podcast, Chain Reaction. Our second episode is about World War I, often referred to as the Chemist's War. I've linked to it in the show notes.
So putting aside his legacy and, the idea that It's almost the art from the artist sort of thing. The Haber-Bosch process is probably one of the areas that has most impacted everyone's life, because 1 in every 2 bites of food that you take comes from a fertilizer that someone made in the lab.
And the Haber-Bosch process is still used today to make fertilizer. It continues to be remarkably cheap and efficient.
So when I say efficient, what I mean is, I think typically on an industrial scale, it's about 97% efficient. So for all of the reagents that you put in to it, almost all of them are going to get converted eventually into ammonia.
And in terms of cost, estimates vary, but Haber-Bosch contributes to around 1 to 2% of the world's total energy costs, and just 3% of carbon dioxide emissions.
The world that we live in today was inherently shaped by the advent of the Haber-Bosch process. It is a modern miracle that it was so right then. So a good analogy might be the Wright brothers first flew at Kitty Hawk in the early 1900s. So imagine if their first plane, essentially, give them a few years to work out the kinks, but by, say, 1914, if that plane was so perfect, we were still using that plane today. Like, that's the level of how right they got it. It's astonishing.
Okay, so we've had the process of making fertilizer down for over a century. It's remarkable, it's essential, but also you can always have too much of a good thing.
In my current job, I'm no longer a bench chemist, but I do work in environmental policy, specifically with regard to water quality. And one of the big challenges that I'm facing in that space, that we're all facing, is something called nitrate pollution, which is when you have fertilizers that are not entirely used by the plants, and then through agricultural runoff, they end up going into bodies of water. And those fertilizers then cause algal blooms, which are fine for the algae, right? They're eating the fertilizer up, but that means that you have dead zones and you have areas where wildlife can't survive. So it's sort of a byproduct of the Haber-Bosch process. So I share this because I was maybe 2 years into my PhD studying dinitrogen fixation, trying to make catalysts that made the fixation process at room temperatures and pressures, trying to find ways to make it with fewer pernicious byproducts. I was writing grants, I was writing articles, and they all started with, like, the single most important thing we can do for humanity is fix nitrogen better. And that's maybe true. But one day I stumbled upon this article maybe it was in the Washington Post or the Times or something, and it was talking about nitrate pollution. And I had never heard of it before. And it was, if you'll forgive the pun, it was a watershed moment for me, because here is this immediate ramification of the kind of work that I'm doing, where on one side, we're talking about how can we fix nitrogen more efficiently, more effectively? How can we create this product that will be used to save people's lives and to allow us all to eat, and to whatever degree you want to defend yourself or create, these economies for pharmaceuticals and for other products. And then on the other side is, again, this immediate policy question of how do we apply it to agricultural fields? How do we deal with the runoff? What are the implications of using this in the real world?
And here's where we get back to the Rich Earth Institute. A lot of their work is based on the fact that we have an overflow of nutrients in water. And in densely populated areas, the main causes of nitrogen pollution are actually septic systems or central sewer plants, which are basically collecting our nitrogen-filled waste into wastewater. So what if there were a way to stop those nutrients, particularly the nitrogen, from getting dumped into wastewater? What if we could reuse them?
Urine has been used as fertilizer by people in their own gardens in many different cultures.
That's Abe Noe Hayes again.
We've heard from folks that, their grandparent in whatever the old country was used to do that as well. But that's, saving urine and using it as fertilizer, which, is still a more common than we realize practice. People tell us, oh yeah, I do this. And it's something that the World Health Organization describes how you can do that safely with your own family's urine without any advanced treatment methods.
If you want to check them out, we've linked to the World Health Organization's guidelines on this.
The idea of using urine in fertilizer or like refining urine to make fertilizer is something the Rich Earth Institute is involved in research on, and that other academics and businesses are doing research on now. I had been doing 10 years of designing composting toilets and servicing composting toilets because I really wanted to recycle nutrients somehow or other, and that was one method. But I was frustrated that there wasn't a legal way to actually return the compost to agriculture. And so I got more focused on urine.
The Rich Earth Institute was founded in 2012 in Brattleboro, Vermont by Abe Noe Hayes and Kim Nace. It was established to advance human urine recycling as a sustainable, locally produced fertilizer, becoming the first community-scale urine nutrient reclamation program in the United States. But in Europe, the kernels of that idea began popping up decades before. In the early 1990s, the first urine-separating toilets were produced in Sweden with the goal of closing the nutrient cycle, reducing the burden on sewage treatment plants.
But these toilets were primarily installed in niche areas, like sustainability-focused eco-villages and vacation homes. For example, some of the early designs were focused on improving the toilet facilities in remote, off-grid summer cottages in Sweden. But they were faced with infrastructure issues.
And that's what the Rich Earth Institute grew out of. I was reading those reports of research and of small pilot operations and thinking, why can't we do that here? Let's do that in the United States. Let's see what it takes to do a community scale project. Because these other projects were more like building scale or laboratory scale, but I wanted something that a whole community could participate in. Anyone in the region could be involved, and that it could be a platform for doing science on the practical logistic aspects of it, as well as the chemical micropollutants from pharmaceuticals or other chemicals, or agronomic effects. Basically, I wanted to build a urine recycling program to show that it was possible and to learn what the obstacles were into doing that, and then how to overcome those obstacles.
One of the first things they needed was to recruit donors.
It was always a funny conversation at the beginning, but then we realized once we started explaining how nutrient cycling works and how humans can be part of it, and how our urine is so valuable as a fertilizer but so polluting as a wastewater, people got it and they're like, how do I join in? And so we established this urine collection program. We got some small grants for research, and that grew into some larger partnerships with universities. University of Michigan, University at Buffalo, and Cornell University have all been close partners in very academic, technical urine research.
And since 2012, it's grown both in scale and operations.
One of our first ways that we collected urine was people manually bringing their sealed jugs of urine to a centralized location. It is vacuum pumped out of their container into our larger storage unit. And as the community fills that up, we then come empty it out and transport it to our research center, where it is treated and pasteurized and prepared for future application with local farms.
Over the years, they've also started installing different toilets in the community that separate solids and liquids, composting the solids and collecting the urine to be used downstream. And Greg told us they have various permitted systems they install in people's homes or at commercial spaces.
For a number of houses, we pull up very similar to people's experience with an oil truck, and there's a port in their house on the side that we run a hose to, and instead of pouring oil in, if you will, we're pulling people's urine out of their basement, often in a big tote and big tank, and that comes to our research center here, where it's treated in a variety of fashions, and pasteurization is the common way that all urine goes through that we process. And then we have a number of farm partners in the region where that urine then goes right back out to the same community, and we apply it with farmers on their land.
Although urine itself is quite low risk, as Greg mentioned, it's treated using pasteurization.
So we always have to assume that the urine that we collect from our 13,000 gallon per year program could have some fecal pathogens in it. So we pasteurize that as the way of destroying any pathogens that might be there. And then that is the same process that is used in treating actual wastewater products. So it's been proven to be effective at much more pathogen-loaded materials. And then at that point, we can sell it, give it away to farmers, gardeners, the public. and then they can use it directly as a liquid fertilizer. We also have ways that we can process that further in order to improve its utility. So one thing that we can do is we can freeze concentrate it.
Rich Earth Institute developed a urine freeze concentrator that concentrates the nutrients in urine, making it easier to store and transport and less labor-intensive to apply.
Freeze concentration is a very old method for taking water out of any liquid solution. People have used it in maple sugaring. If the sap freezes in the bucket on the tree, you can pull out the ice and throw it away because it's just pure water ice. To make applejack, you take hard cider and you partially freeze that, remove the ice, and then what's left is a more concentrated alcohol. So the principle is that when an aqueous solution freezes, the ice is a pure water structure. It's a water crystal. And so all the solutes are excluded from that crystal, and they're still in the liquid water fraction. So if you are able to separate the ice from the remaining liquid, then you have a concentrated liquid, and you have ice that you can melt and get pure water from.
Rich Earth Institute created the spin-off company Brightwater Tools, where they sell the freeze concentrator and other technologies and equipment for urine processing. But a lot of what the Rich Earth Institute does as a nonprofit is research, including looking at what's in the urine they're collecting.
Abe told us the contaminants that have come up in this project have been various pathogens, heavy metals, pharmaceuticals, and PFAS, aka forever chemicals, a topic that we just cannot get away from on Tiny Matters. So pathogens are handled in the heat treatment process of pasteurization, and fortunately, the level of heavy metals is so low that no additional treatment has been required. Then there are pharmaceuticals.
We did many years of field trials looking at the pharmaceuticals in urine, what would happen to them when you applied them to agricultural soil. And that had a very encouraging result, because we found that even with very sophisticated detection methods with mass spectrometry, there was no detection of most of the pharmaceuticals we looked at. What we did find were things like caffeine or over-the-counter pain medicines, but the levels were so small that in order to get a single therapeutic dose of any of those drugs, like say the amount of caffeine in a cup of coffee, you'd have to eat an entire salad from this study plot for about 2,000 years before you would accumulate that.
Okay, so no concerns there. Abe told us that every year they're actually required to test for different PFAS compounds. You might be comforted to know they're also finding those at very low levels.
The one that can be detected with any kind of regularity is PFBA. It's one of the shortest chain ones. And the levels that it is detected at are quite low compared to other materials that are used in agriculture. And so we're working on a paper now, actually, that will put that into context with other materials and to try to show the relative amounts of PFAS that are going to farmland from different sources. And urine is on the very low end of that spectrum.
In addition to testing, a lot of what drives the work at the Rich Earth Institute and Brightwater Tools is the need to manage the nutrients in the wastewater, which we talked about with Leila. Fertilizer helps feed billions of people, but on the flip side, there's now so much of it that we're dealing with nitrate pollution from both agricultural runoff and wastewater. So Rich Earth Institute is working with various towns in New England, consulting on the social and the practical aspects of setting up urine diversion for a community so that they can avoid wildly expensive sewage and septic costs that come with cleanups.
Nutrient removal systems are fantastically expensive. And if we can just divert most of those nutrients before they even get in our wastewater, we've solved most of the problem. Realizing that you can collect 80% of that nitrogen just by putting your urine into a collection system, that's a hugely powerful tool for communities that are having an existential crisis. And if we can help provide a tool to help them not only solve their problem, but also turn what comes out of their body into a resource, it feels very important. That's part of what motivates me so much on this.
For the future of urine recycling, I think that it is realistic that these systems will start to intersect with people's lives more and more without them potentially being aware. You might go to the bathroom in a urinal or in some public space and not realize that there's a collection system that is plumbed into and that is part of a local circular economy to return those nutrients to the land. But idealistically, I would love to see just a consciousness about waste in general, but particularly human waste, as it's a thing that we all have a very direct relationship and an awareness of that we do every day.
I feel like as we increasingly learn more about the human impact on our environment and climate change, this idea that we need to be more aware of the systems that make up our lives is a sentiment that I feel like I'm seeing more and more in the people we talk with for Tiny Matters.
Right. Even something as seemingly simple as pee could make a huge difference in how our world functions, and that's shaped by both individual and collective changes.
All right, Deboki, tiny show and tell time.
Sure. I think I can go. I think last time
All
I
right.
might have gone first. I don't know.
I feel like yes, but I also don't know. Oh no, our last episode was with distillations, and so we didn't actually do a tiny show and tell at the end.
Well,
But then before that, it was me. So yes, you can go.
Cool. Well, I have been granted permission to go. So for my tiny show and tell, I want to talk about how scientists make viruses better at fighting bacteria, which is like not necessarily the goal of this project, but it was sort of what I decided to take from it. So the way that you do this is you send them into space.
What?
Yeah. Okay. So the viruses that we're going to be talking about are bacteriophages, which specifically, infect target bacteria.
Yes.
Specifically, these scientists looked at a bacteriophage called T7 that's used in a ton of labs, along with E. coli. They had a batch here on Earth. They also had a batch up in the International Space Station, and they looked at how these viruses were infecting the bacteria.
The E. coli.
Yeah, sorry. Yeah, the
Okay.
E. coli. So on Earth, it took about 2 to 4 hours to infect, and the ones in space took about 4 hours. So on first glance, I saw that and I was like, that doesn't actually seem like it's better. But the thing is that after the viruses were adapted to space, it seems like they were actually really good at killing the bacteria. And so what the researchers think was happening is that basically in space, because of like the lack of gravity or because they're in microgravity, the bacteriophage weren't mixing as well with the bacteria. So it was like harder for them to find their targets. So they also had to like adapt to this new situation. It was like stressful for the bacteria. It's stressful for the viruses.
I'm just picturing the viruses and bacteria like, whoa, it's so crazy up here.
Exactly. That is what is happening. But after the viruses adapted, they had mutations that like changed their shape and made them better at grabbing onto bacteria. So they were like good at killing the bacteria.
Wait, why? Sorry, why do they think that is? Are they trying to figure it out?
I think it's because of the adaptations they needed specifically to get like adapted to microgravity.
Oh.
Yeah. So like maybe in that situation, you're selecting for bacteriophage that are like really like have a certain shape, have a certain capacity for targeting the bacteria and like are able to grab onto them. And so like they're the ones that are going to keep, surviving and thriving.
Got it.
Right? So one of the interesting potential applications, which I hadn't thought about, but it was wild to me that they were saying this, is the idea that growing bacteriophage in a new environment under new stressful conditions could like make them better at treating treatment-resistant bacteria. And I was like,
I like
weird.
that.
What if we just started sending bacteriophage into space so that we could deal with treatment-resistant bacteria? I think we would just grow them in like microgravity like chambers or something.
We probably
that's probably
don't need to send them to the ISS every
Yeah.
time. We're like, all right, get these guys ready to go.
Yeah. So, I mean, obviously, I don't know if that's going to be what like the actual application of these results are, but I just thought that was really fascinating.
That is really fascinating. It also, though, makes my brain go in like a more anxious direction of viruses specifically being able to adapt really well in those kinds of environments, and thinking about things like SARS-CoV-2 or other, could they become more effective, not just to kill off bacteria, but could they infect human cells more effectively? I don't know, but I don't think we should be testing that in the ISS yet.
No, probably not.
Let's not.
Which I'm guessing is why they used bacteriophage and bacteria and not the ones that get us.
Right.
No, but I think that's like a really great point because we don't really know like how these different microbes are going to adapt to different situations. And every time we send people into space, we're like learning both through experiments and also kind of through just like the fact that people are now in space and their microbes are there. And
there's only so much you can control for, especially when like every surface of your body has microbes all over it.
This like actually reminds me way back when we did
The moon episode.
the moon bacteria and like coming back, like and the fear that there was going to be like bacteria from the moon or like infections from the moon,
and how they set up a station that was just like could not have had more issues. Where if there had been an issue, if they had brought something back that was harmful to humanity, we would have been in a
terrible
bad spot.
Yeah.
Yeah. Okay. So interesting. That is fascinating.
Yeah.
Wow. Okay. I love that. Thank you, Deboki. So I'm going to talk about dead stuff because what? Because I always am. I want to talk about mummies, specifically mummy smell. And I know that I've talked about this before. I know that I brought this topic up in the past. I was trying to figure it out. I think it was in a tiny show and tell sometime a little over a year ago. But also we did a mummification episode for Halloween a while ago. So like this concept has come up. But this is a big improvement in what researchers have learned and what it could mean. And I just think it's really interesting.
Cool.
Okay, so there's this new research looking at mummified remains in more detail to get an understanding of how elaborate burial rituals were. So this is led by chemists at the University of Bristol. It was published in the Journal of Archaeological Science. And what they did was they analyzed 35 samples of bombs and bandages from 19 different mummies. And the mummies, age-wise, ranged from 3,200 BCE. That's a long time ago.
Yeah.
It's hard to conceptualize. To 395 CE.
I think like at least like every other month in our household, we have a moment of like realizing both like how long ago like Egyptians were around and how long.
Oh, totally. What was so interesting is that in these 19 mummies, researchers identified 81 distinct VOCs or volatile organic compounds. So these are like compounds that are, I think of like wafting off of.
Right.
Obviously, the analysis was it was really amazing. The technology they used was just so advanced that they only had to take, I think they said like a peppercorn-sized fragment from these mummies to be able to analyze the different VOCs, which I thought was just really cool. Because of course, that's like one of the things when you're trying to analyze ancient samples of any sort is you don't want to destroy things inadvertently, right? Like, so I think that's why I think I've talked about in the past too, like that there's someone who figured out how to do CT scans for mummies so you don't have to fully unwrap their bandages to be able to see, and that's how they figured out that one of the ancient Egyptian rulers was actually murdered and didn't die of natural causes because of something. I'm getting away from smells. Let's get back to how these smell. Okay, so it was interesting because not only did they figure out that the chemical patterns of scent, so like the different VOCs, were varying across historical periods, which is like, okay, yeah, that makes sense. They also figured out that they varied depending on what part of the body was sampled. So there's different things going on here. So first off, just going back in terms of the variation over time, earlier mummies, so according to the first author, Dr. Guano Wars, they found that earlier mummies had these more simple profiles, was how he said it in his quote, dominated by fats and oils. But then later, they had these more complex mixtures. So they had like resins, bitumen, I always forget, but it's like asphalt, tar. So you have this strong like petroleum pungent odor. And then I think they were finding more like woody, earthy, balsamic odors. So that was really interesting because, over the course of time, you're seeing that different techniques are used and different compounds are used in this embalming process. So that's cool. But then depending on the part of the body, that also changed. And so that I thought was really interesting because it would infer that, embalmers were using different recipes for different parts of the body to aid in preservation. Like thinking about, okay, so like obviously the way that a part of your body, like your foot would decompose, is very different to like your head, which stores your brain. And, like there's a lot of oozy stuff going
right.
on. So it's just really interesting to see that like being able to detect these VOCs is not just cool to look at, over the course of time, how what they used varied, but just thinking about like how really advanced their knowledge was at the time.
Right.
About, thousands of years ago about decomposition.
Yeah.
Right? Like what was happening to a corpse or to different organs or parts of the body as it decomposed, and then varying their embalming process based on that. Like, I think that's just cool to think about,
Yeah. That's really fascinating. And it's so interesting to realize like how much they must have known. Like, and it makes sense. Like, it's all like a thing you can observe, I guess. Like
we think we're so smart and like, okay, we're smart as a species, but how much smarter are we now? Debatable.
Yeah. Yeah. Oh, wow. That's really cool and very on brand. Yeah. And one day we'll just have the Sam mummy episode that's just more and more of these.
I know, truly. I feel like I need to surprise you at some point, Deboki, by not doing something related to like death, disease, pestilence.
Yeah.
One of these days.
I like the idea. Listeners, I would like to issue a challenge for whoever can like send us a death fact or like a dead body fact.
Yeah.
That is like the most shocking to Sam, the most new thing to Sam.
Yeah. Make it the most Sam-coded fact you can imagine.
Right.
And I'll send you an email with my undying gratitude.
Yeah. Well, thank you, Sam. And thanks to all of you for tuning in to this week's episode of Tiny Matters, a podcast brought to you by the American Chemical Society and produced by Multitude. This week's script was written by Sam, who is also our exec producer, and edited by Michael David and by me, Deboki Chakravarti. It was fact-checked by Michelle Boucher. Our audio engineer is Misha Stanton. The Tiny Matters theme is by Michael Simonelli and the Charts and Leisure team.
Thanks so much to Leila Duman, Abe Noe Hayes, and Greg Riley for joining us. If you have an episode idea, email us, tinymatters@acs.org. We'll see you next time.